Hello everyone, this is Venkatesh, first MSc Microbiology from Sacred College, Tirupattur. In this video lecture, we are going to discuss about biosynthesis of purines. A basic introduction about purine biosynthesis. In general, the purine and pyrimidine that are considering as a nitrogenous base which contribute for the nucleic acid synthesis. The purine that are 2 carbon nitrogen containing ring base, which means this is considering as the pyrimidine base pair and this is considering as the imidazole ring which can be found in the nucleotide which make up the DNA and RNA. So in simple term we call it as nucleotide are the backbone of nucleic acid, and then purines are water soluble and weak base in nature. So what are the sources that are required for the synthesis of purine? So the first major source is the sugar molecule, that is the ribose, which is a 5 carbon atom that are derived from carbohydrate metabolism, either pentose phosphate pathway or Entner-Duderov pathway. The secondly, certain amino acids like aspartic acid, glycine and glutamine, which are the intermediates that are produced during the glycolysis and Krebs cycle that are going to participate in the biosynthesis of purine. Here I have listed out some of the chemical elements which contributes to make up the purine ring structure. So where the purine biosynthesis taking place means liver is the major site for the synthesis of purine. Purine biosynthesis pathways. There are two pathways for the synthesis of purine nucleotides. The first one is de novo pathway. It's a biochemical pathway for the synthesis of purine nucleotide from simple precursor molecules. The second, salvage pathway, which is used to recover the purine bases and from the degradation of nucleic acids such as DNA and RNA. Let's we have a closer look on purine synthesis pathway. Normally, ribose sugar is inactive form. It has to be activated for the synthesis of purine nucleotide by adding the phosphate group in the fifth position of the ribose sugar. This can be done through carbohydrate metabolism of EMP pathway or pentose phosphate pathway. Here we have the ribose phosphate sugar, which is active form that reacts with the ATP to form phosphoribosyl pyrophosphate. So which is catalyzed by the enzyme called PRP synthetase enzyme. That is, the energy utilization process is taking place, whereas the ATP is reduced into AMP, which provides its 2-phosphate group to PRPPV substrate in the first position of the ribose sugar. In the second step, the glutamine amino acid that transfer its amine group of the PRPP and become reduced into glutamate to form the 5-phosphoribyl amine, which is catalyzed by the enzyme called PRPP glutamine amidinotransferase. In the third step, phosphoribosyl amine that reacts with the glycine in the presence of ATP that forms glycinamide ribonucleotide. Here the energy utilization process is taking place, which is catalyzed by the GAR synthetase enzyme. In the fourth step, as I mentioned before, there are 2 rings is present in the purine nucleotide. From fourth step onward, the imidazole ring is formed. So here a chemical compound called THF, which means the tetrahydrofolate, which is a purine derivative and also known as vitamin B9, which is folic acid, that used to bind with the chemical compound which is having the 1 carbon in its structure to donate formyl and methyl groups in the anabolism process. So here the N10 formyl tetrahydrofolate donates its formyl group to form formyl glycinamide ribonucleotide, which is catalyzed by the enzyme called GAR transformylase. In the fifth step, the amino acid glutamine get reduced into glutamate, which again transfer the amide group to form formyl glycinamide ribonucleotide, which is catalyzed by the enzyme synthetase. Again the energy utilization process is taking, whereas the ATPs get reduced into ADP plus inorganic phosphate. In the sixth step, The imidazole ring of the purine is closed in an ATP-dependent reaction to yield 5-aminoimidazole ribonucleotide, which is catalyzed by the enzyme called synthetase. Again, the energy utilization process is taking place. In the seventh step, the incorporation of CO2 occurs in the C4 position of the imidazole ring that leads to the formation of carboxyaminoimidazole ribonucleotide, which is catalyzed the enzyme called carboxylase. In this step, the aspartate amino acid that contains with the carboxyaminoimidazole ribonucleotide to form aminoimidazole 4-succinyl carboxamide ribonucleotide, which is catalyzed by the enzyme called synthetase. Again, the energy utilization process is taking place. So in the ninth step, the enzyme called adenosuccinate lyase that cleaves off the fumarate. This is fumarate, and only the amino group, which is aspartate of the amino group, that is retained to form aminoimidazole 4-carboxamide ribonucleotide. In this step, again, the N10-formyl tetrahydrofolate, which is the folic acid derivative that transfer its formyl group to yield 5-formyl aminoimidazole 4-carboxamide ribonucleotide, which is catalyzed by the enzyme called transformylase enzyme. So in the 11th step, the enzyme called IMP synthase, which leads to enclosing the imidazole ring with the elimination of water. The product obtained is inosine monophosphate, which is the parent purine nucleotide from which the other nucleotide can be synthesized. So here, inosine monophosphate is the immediate precursor for the formation of AMP as well as the GMP. The amino acid aspartate that contains with the inosine monophosphate that leads to yield the adenylosuccinate substrate, which is catalyzed by the enzyme called adenylosuccinate synthetase. Here the energy utilization process is taking place, whereas the GTP is reduced to GDP plus inorganic phosphate. Here the enzyme called adenylosuccinate that cleave off the fumarate from the adenylosuccinate and retain only the amide group that leads to form the adenosine monophosphate. In other hand, the inosine monophosphate that undergo NAD plus dependent dehydrogenation process, which leads to yield xanthosine monophosphate. Here the amino acid glutamine that get reduced to glutamate by donating its amide group to xanthosine monophosphate to form guanosine monophosphate. Hence, we could conclude the inosine monophosphate which act as a immediate precursor for the formation of AMP, which is adenosine monophosphate, as well as the guanosine monophosphate, which is the nucleotide base of purine. With this, the biosynthesis of purine is explained. So thanks for your pleasant listening and catch you on next interesting topic. Thank you.